So, hello everyone. My name is Clinton. Um, I studied at Carnegie Mellon University for my bachelor's degree in biological sciences. I did an honors degree there, and I also did two master's degrees, one in molecular medicine, that's wet lab research, and another one in dry lab research, so bioinformatics. Um, I've learned several skills along the way, such as Python, R, Linux programming, and I've won some awards in different institutions. I also have a software engineering diploma, and I love both biology and programming. So, just a little bit about my background. My first master's was about studying a protein known as ST20 like kinase, or SLK. And this was in a cancer lab where we studied different cell lines, some mouse models. Essentially, we were working on characterizing SLK's role in apoptosis, cell cycle, and cell migration, some common cancer phenotypes. And some things that we found out were that it's critical in normal development and a complete knockout's lethal. And it plays a role in new induced mammary tumorigenesis. So breast cancer in mice. So that could have a phenotype, a human phenotype for future projects. So these are just some results. If you're used to wet lab techniques, you might be familiar with Western blots and focal adhesion assays. So the aim was to find out what SLK the role of SLK or ST20-like kinase in mouse embryonic fibroblasts by testing common cancer processes such as proliferation, migration, and apoptosis. And finally, the result was that we noticed that focal adhesion turnover seems to be impaired. So I studied this at the University of Ottawa, where I did my first master's. So after doing my wet lab research within a cancer lab, I I felt like I wanted to learn more analytical skills to analyze my data set. So looking at the trend around me, I noticed that biology was heading towards a more data science approach, more where you have to analyze different types of data with more sophisticated software tools. So then I realized that let me transition to a more bioinformatics role, and I joined another master's immediately after my first um, in the University of Guelph. So I did a master's in bioinformatics, and some of the reasons why I did that was because of the changing dynamics of the field, my passion for both biology and programming. I did get into computer science at Carnegie Mellon for my bachelor's, but at the time I was more interested in biology, and I never really knew much about bioinformatics or computational biology. So I because of that, I got exposed to different disciplines such as statistics, mathematics, engineering, computer science, something that a person who just did biology might not generally be exposed to. So it did give me a lot of variety, and I did enjoy that. So it was very, and I still enjoy it. So after doing my master's in bioinformatics, um, I met Ilya through one of his interesting webinars, and I he was talking about his bioinformatics for precision oncology program, and we were discussing about the different aspects, and I had the opportunity to be one of the mentors for the program. I'm currently doing a teaching and training of adult certificate expected in August. So I do have the formal teaching skills to embark in such a program. And it was a very nice experience because I have the interest for cancer research, and it was very enlightening to see my students with their own research coming from a more wet lab approach, which is something I'm familiar with. So it was a very I had the opportunity to talk to um, wet lab research scientists, give them my insights, use the resources that we have to help improve their research by incorporating important data science techniques. But for this program, it was particularly data science techniques for cancer research. But um, the program that we're talking about is more the important software skills for more generic types of research. So these techniques are very important. And being a person who transitioned from a wet lab approach to a dry lab approach, I can definitely appreciate those techniques. And this was a fun experience, and I look forward to repeating the same with you all. So that is, does anyone have any questions?
So Clinton, I actually do have a question for you. I'll represent the audience here. So my question is, how difficult was it to transition from learning about life sciences and biology to data science, or to, other things that you've done?
So initially, I thought it might be very difficult because there were some programming concepts like R was a bit new to me. I did a little bit of Python I did some Python in my bachelor's degree, but it was minimal. I think with every new experience, you kind of there's a learning curve. So no matter what you do, you will have some sort of learning curve, and most people have to go through it. Thankfully, being in a good environment, so being in this course, you'll have experienced mentors to give you that learning environment, just like my professors during my master's program. So I'd say in my experience, every challenge has a learning curve. It's preferable to have a good learning environment and good mentors to help you support you during that learning curve.
And another question that I wanted to ask was, how do you know when you've learned something? I mean, there are many resources online, right? You can just download people's code and, try it and things like that. I mean, how do you know that you've actually learned some analytical technique or method?
So, because I am in the process, and I have been an instructor, there is a taxonomy. It's called Bloom's taxonomy. So there are different levels of learning. So it depends on how much depth you want to go into. So you do have a point that sometimes you might not be sure if you learned something. And educators like to use concept checking questions, different types of assessment, whether it's coding assessments or just simple quizzes or multiple choice questions. It's usually good to display your work in a project of some sort. So that really shows that you've mastered the tiny puzzle pieces in order to create your own novel work. So that would be one of the higher levels on that taxonomy versus just typing a print command, which just kind of shows you that you memorized the recall. So you have that recall stage. So ultimately demonstrating your work in a creative project, that would show like your ultimate understanding. So through a research project. So whether that's publishing or doing presentations in front of other experienced people, or creating some manuscript, or doing a poster. So yeah.
Okay, great. Thank you very much. And if you do have any other questions, please do write them in the chat box and we'll be sure to address those. And I briefly want